so we can hopefully keep to our schedule. So as you just heard, these talks are being recorded. And so, yeah, so welcome to the CSAFM Observational Studies of Land Atmosphere Interactions Part 1. So it's great to see you all here today. We have an exciting lineup of talks. So we'll have 3 15-minute talks, and then we'll have 2 quick poster presentations, 5 minutes each, and then we'll have another set of 3 15-minute talks to end the session here today. And just as a reminder to our speakers, just a note that talks are 15 minutes, so try to keep your talk to 12 minutes so that we can have about 3 minutes for some Q&A at the end. So with that, let's go ahead and get started. So our first speaker is Jacob Evans from Guelph, and take it away.
All right, good afternoon. My name is Jacob Evans, and I'm a graduate from the University of Guelph. I'll be presenting part of my study, which was about carbon exchange altered by crop diversification and the use of cover crops. I hope everyone can hear me clearly. Okay, so so there's the recognition that agriculture can help in the fight against climate change through net carbon uptake. And so this system, in this slide, I wanted to highlight the role of the role of plant in carbon exchange. So basically, if we consider this system, the presence of plant allows for photosynthesis to move carbon into the system. At the same time, we have respiration occurring in the soil and in the plant, which moves carbon out of the system, so that the term gross primary productivity, GPP for short, is used to describe the photosynthesis occurring in this system. And the term ecosystem respiration, RE for short, is used to describe the total respiration occurring in this system, such that the difference between these two systems is what we refer to as the net ecosystem exchange, NEE for short. Well, what this basically means is that if we have a transfer of carbon out of the system, we say it's a loss, and that basically means that respiration is larger than GPP, and vice versa. I mean, if we have a net transfer of carbon into the surface, we say that it's a gain, carbon gain. Okay, so we include an additional term, the net ecosystem carbon budget, NECB for short, to describe carbon that is lost, to account for carbon that is lost via plant harvest. So what are we basically saying, in essence, in a nutshell, is that the presence or absence of plant or vegetative cover would significantly influence carbon exchange dynamics. And so if we consider the predominant cropping system in Ontario, the corn, soybean, we see that crop is usually grown for about a short period of time, say about 4 or 5 months, and we have a long fallow period, which corresponds to carbon loss. And so this reveals the need for more management options, improved management options, basically, that can, increase carbon inputs to soil, which is what my study is basically all about. So that the objective for this study, number one, is basically to compare the seasonal dynamics of any GPP and respiration between diverse and non-diverse rotations, and number two, to determine if diverse and non-diverse rotations are net ecosystem carbon losses or carbon gains. So I delve into the methods. I conducted this research, this study at the Elora Research Station. At this site, I had 1 8-hectare plot cropped to non-diverse rotation, and the adjacent 8-hectare plot cropped to diverse rotation, and eddy covariance was installed in each of these plots side by side. And here is a picture of the instrument used to measure the NEA flux. Like I said, I had each of these installed in each of the plots, and each tower basically comprised of a 3D sonic anemometer and a CO2 gas analyzer, which was used to measure the NEA flux at a frequency of 10 Hertz. And this high frequency flux was then averaged to 30 minutes time series using Eddy Pro, and thereafter partitioned to respiration and GPP. And then I invoked gap filling algorithm to basically obtain complete annual data sets. And then I also created 50 artificial gaps scenarios, basically to derive error terms for NEE, respiration, and GPP, which I kind of use to compare between treatment. Okay, so to address the second objective, which was about the net ecosystem carbon budget, I sampled for plant yield. I sampled plant for yield at the end of the growing season and oven dried it and sent it to the lab for carbon content analysis. So let's delve right into the result. The cropping sequence for this study is such that, like I said, the non-diverse treatment, which is the corn, soybean rotation, the predominant rotation in Ontario. And then I diversified this rotation to include a 2 cover crop mix, such as crimson clover and then ryegrass in the first year underseeded with corn. In the second year after soybean harvest, winter wheat was sown to keep the ground cover out through the winter. And after it harvests in the third year, a 4 mixed cover crop, which included crimson clover, cereal, rye, radish, and oats, was sown. Okay, so for my first objective, which I wanted to compare the NEE, RE, and GPP dynamics between non-diverse and diverse rotations for each study year, which is a period between May and April for each year. We see the cumulative values for the first year, which is the corn year. The bottom graph here shows the cumulative NE, and the other two shows the partition. Remember, we said that NE is composed of RE and GPP. Okay, so the green and the red curves here basically shows the diverse and the non-diverse treatment respectively, and the vertical dashed line here shows when plants were either sown or harvested. The gray lines you see visible in some, not visible in some, are the 50 artificial gap field scenarios I talked about earlier, so that the deviation of these numbers gives the error term here that is used for comparison between the treatments. Okay, so again, the sign convention I use in this study is such that, especially for those not familiar with this kind of trend, when the NEE is increasing into the positive territory, we say it's a carbon loss. When it is decreasing into the negative territory, we say it's a net carbon gain. And when it is flat, it basically means there's not much happening there. The GPP is negative because, again, that's the uptake component, and the respiration is positive because that's the loss component. So at the end of the first year, the net is negative, and this basically goes to show that GPP was larger than the respiration, for both fields. And this is kind of what we expected, because corn is a C4 plant, and as such has maximum photosynthetic efficiency. Aside that, we see difference in the GPP between the two treatments. And this is, again, because we had cover crop growing in the diverse field, right? Which was also accompanied by increased respiration in the diverse field as well. And the difference between these two, eventually we do not see a difference in the NE, because, again, the NE is basically the difference between the respiration and GPP, so that we do not see a difference between the two treatments in the NE section. Yeah. So I show a picture of corn, in October, showing corn dried down in both fields, diverse and the non-diverse field, with cover crops in the diverse field. You see nicely here growing. In the second year, it's also presented as the same format in the first year, but the magnitude of NE is different from the first year. The NE was positive here at the end, because respiration was larger than GPP, which is kind of what we expected, because soybean is a C3 plant, right? And the trend was very similar. The NE trend was very similar, because this is kind of, again, what we expected, because both fields were managed identically. Again, after soybean harvest, we had wheat planting, and this kind of increased GPP uptake in the diverse field, accompanied also by an increase in the respiration in the diverse field. And eventually, again, we do not see a difference in the net. Okay, so this is also a picture taken October 15th, showing, after soybean harvest, showing winter wheat beginning to emerge faintly in the diverse fields. Okay, so it gets interesting in the third year, the pattern is different this time around between the two NE curves because the timing of things was basically different. Wheat sown in the previous year, together with cover crop sown in this current year, decreased carbon loss that would have otherwise occurred in this shoulder season. And this is because they begin to take up carbon in this period, such that at the end, GPP was significantly higher in the diverse treatment, which was accompanied by moderate increase also in respiration, leading to more carbon gains in the diverse fields at the end when we look at the net, right? So yeah, here's a picture showing both fields in July before wheat harvest. Side by side, you see the soybean in the non-diverse field, and yeah, after wheat harvest, and yeah, after the cover crop had been established in October. Okay, so then is diverse all good, right? I don't know. Okay, well, not really. I wouldn't say I don't know, not really, because when we consider the carbon removed at the end of the carbon removed at harvest, it was not significantly different in the first year, in the second year as well, but the third year was different. There was more carbon removed in the diverse field, so then this cancels that difference we see in NE. So that if we take a look at the NECB, which is again the difference between the NE and the carbon removed, the totals for the 3-year study period revealed that the diverse cultivation was a higher net carbon loss compared to the net, compared to the non-diverse rotation. So in summary, basically we see that from the net carbon budget, the amount of carbon exported from fields can basically override the ability of fields to act as either carbon losses or gains. So in conclusion, from my study, I did see good agreement between measurement in adjacent field as indicated in the second year. And the active growing season, the diverse rotation, especially in the third year, did increase the potential to act as carbon sink to some extent, but overall it was not enough for this study period I considered. And then this result can only suggest, likely trends that are reflected in long-term carbon balance, resulting from sound management practices in agricultural systems. Okay, so I'd like to appreciate the AgMet lab under the supervision of Claudia. I believe she's present here today, and also for being a beneficiary of the Create Climate Smart Soils program. All right, thank you for listening. I'll be happy to take any question.
Wonderful. Thank you, Jacob, for that nice talk. So we can put questions in the chat. I'll read them out loud. We have time for a couple questions. And feel free to put your questions in the chat as the talks are ongoing. All right, so from Catherine, do you think you would have seen a greater difference if different cover crops were used or different rotations? Oh, please unmute yourself, Jacob.
Yeah, hello, sorry about that. Okay, so if we had used different cover crops or different rotation, well, it depends on what kind of cover crops we're talking about here. If the cover crops, if we had rotations that would have to, pull, do that exchange more efficiently, then perhaps, yes, we could see even more uptake of this. Like I said, our treatment was basically the corn, soybean, and we just basically wanted to focus on this predominant rotation and see how we could diversify this. And yes, I think, yeah, if we do use some other cover crops that has more, photosynthetically efficiency, that could, yes, perhaps in some way impact the dynamics of the carbon trends that we see in my study.
Wonderful. And one last question from Timmy. Can you comment on the weather conditions, wet, dry, compared to historical average in each of the 3 study years?
Okay, so the last year was a bit, that's in 2020, 2021, that's the study year. For the last year, it was a bit wet. I mean, the soybean went into the ground early compared to the first 2 years. The first 2 years were not as wet as compared to the last year, basically. Yeah.
Great, wonderful. Thank you so much. We'll move on to our next talk. So our next talk is by Nicole Menhiri from the University of Guelph. So I will let you go ahead and share your screen. And for the presenters, I'll give you a 2-minute warning when you're at about 11 minutes, just to sort of give you an indication of time. So, all right, thank you.
Awesome. So, hi everyone, my name is Nicole Menhiri. I'm here today speaking about my master's research on mitigating nitrous oxide emissions from corn using nitrification and urease inhibitors following cover crop adoption. So, So when fertilizers are applied to agricultural soils to increase yields, they go through the nitrogen cycle and the processes of the nitrogen cycle. The nitrification reaction converts ammonium into very unstable bioavailable nitrate, which can be taken up by the cash crop, immobilized into the soil, leached into groundwater, or in oxygen-limiting environments, convert into nitrous oxide through the process of denitrification. Nitrous oxide has 300 times the heat trapping capacity of carbon dioxide and can destroy atmospheric ozone. Cover crops can reduce nitrous oxide emissions during freeze-thaw periods in the spring by taking up excess nitrogen and reducing the extent of soil freezing. But after termination, the excess carbon and nitrogen applied to the soil from the cover crops decomposing can actually increase nitrous oxide emissions when synthetic fertilizers are applied. This flush of bioavailable nitrogen accelerates microbial respiration, which quickly makes the soil environment oxygen limiting and allows for incomplete denitrification to happen and a lot of nitrous oxide to be produced. So our proposed solution is, instead of getting rid of cover crops and all the benefits they can bring to your farm, why not combine them with nitrification and urease inhibitors like Tribune? So nitrification and urease inhibitors slow the breakdown of fertilizers in the soil by blocking the active sites of enzymes within nitrifying bacteria and nitrosomonas. So if you recall, the nitrification reaction comes just before the denitrification reaction. So if we can slow that first reaction down, we can maybe slow denitrification down and nitrous oxide production down as well. And also, it's good for the farmers because nitrification and urease inhibitors can extend the window for cash crop uptake. And so rather losing nitrogen to the atmosphere, more nitrogen goes into the crops and can increase yields better. And so nitrification and urease inhibitors have not only shown to reduce nitrous oxide emissions on conventional rotations, but can be economically beneficial by improving nitrogen use efficiency, but they've never been studied combined with cover crops. So that's why I'm here. So I'm just gonna quickly go over our micrometeorological site where we quantify the nitrous oxide flux. And we use the flux gradient method to quantify this nitrous oxide flux. And in order to do that, we need to collect trace gas emissions, which are collected by our intakes in light blue there. We also need to get the concentration of those trace gas emissions at the 2 known heights. And we use a tunable diode trace gas analyzer in purple, and it's located at the center of the plot. And we also, to characterize the turbulence in the atmosphere, we also have a cup tower in orange, and we also have sonic anemometers, one in each plot with different surface roughnesses, and pyranometers and pyrgeometers to measure the incoming and outgoing radiation. We also measure soil temperature and moisture in the yellowish circle there, where we have hygrometers and thermocouples buried at 2 known depths in those circles. And we have other thermocouples and hygrometers buried deeper. And we also collect soil samples for dissolved nitrate and ammonia concentrations and plant samples for plant nitrogen uptake. So now I'm going to go over a quick overview of our 2021 growing season. It started with our 4-away cover crop mixture that Jacob was mentioning with the daikon radish, crimson clover, spring oat, and rye. And we had about 2.7 tons per hectare of cover crop coverage in those 2 plots. And so close to corn planting, we terminated the cover crops chemically and integrated them into the soil before corn was planted. And a couple weeks after corn was planted, it was at the 6 leaf stage and then nitrification and urease inhibitor tribune was applied to the top 2 plots there, one in a conventional and one in a diverse plot. Shortly after, crimson clover and rye into the corn in our diverse plots on the left-hand side there. And a month or so later, our corn was ready. A month or so later, our corn looked like this, had a nice cover crop coverage on our diverse plots, and on our conventional plots, as you can see, there's no cover crops. And here are the yields we got for our plots. So our yields from current statistical analysis, I'm still looking into it, have shown to not have statistically significant results, but we can see slight variations between the plots. So now I'm going to hop on into the results. So this plot here is a plot of our 2021 cumulative nitrous oxide emissions, and this is just highlighting the difference between the conventional and the diverse plot, diverse in green and conventional in yellow, and highlighting the difference between their nitrous oxide emissions during the freeze-thaw period of 2021. As you can see, our conventional plot, we saw a much higher nitrous oxide emission event shortly after freeze-thaw in between March and April there. Now we have daily nitrous oxide emissions, and I've highlighted a couple of big events for the field. So we applied UAN here on the first line, and then we saw a little nitrous oxide emission event there. Then we had rain, and we had another rain day followed by our cover crop interseeding into the corn. And then we saw a very large nitrous oxide emission event. And just a reminder, these are daily emissions. I just switched from cumulative to daily. So just reminding you there, don't need to get confused. And our big difference that we're seeing here is our diverse plots are seeing much higher emissions than our conventional plots, just in that snapshot right after that fertilizer has been applied. And we don't see much of a difference between plots that did or did not see the inhibitors. So here is a plot of our annual cumulative emissions. Again, I have bars there to show big events. So we have our freeze thaw. We also have our cover crop termination, our second bar. Then we have UAN application, our third bar. Our fourth bar is that cover crop planting rain event. And then you can see the large nitrous oxide emissions being produced, mostly from our diverse plots. Our diverse plots saw much higher nitrous oxide emission events at this time, whereas our conventional plots did not. But as you can see, closer to harvest, we have a better idea of what's going on the entire year. And we saw differences basically between the conventional and the diverse plot, we saw differences in cumulative nitrous oxide emissions, but the plots that received nitrification and urease inhibitors, regardless of their cropping rotation, had pretty similar cumulative nitrous oxide emissions. So as you can see, our conventional NUIs in our conventional plots reduced nitrous oxide emissions by 7.4%. And in our diverse plots, the NUI tribune reduced nitrous oxide emissions by 20.6%. And the cumulative nitrous oxide emissions give you the daily are interesting, they're showy, they're kind of dramatic, but the cumulative nitrous oxide emissions are where we're really seeing the results of the whole year. So we have some initial conclusions. I still have some gap filling and some statistical analysis to do, but our conventional fields emitted more nitrous oxide during the spring freeze-thaw period in 2021, but our diverse fields emitted more nitrous oxide after synthetic fertilizer was applied in 2021. And the fields that received nitrification and urease inhibitors had lower annual cumulative nitrous oxide emissions, regardless of their regardless of their cropping practice. Cover cropping, it's hard. So our research impact, the big why of what we're doing here is we really want to inform emission factors, which help governmental bodies track national greenhouse gas emissions. As a lot of you may heard, the Canadian government just released the new climate plan, which has put a cap on agricultural emissions 30% below the 2020 level of emissions that we saw. So in order to do this, we really need to look at combining best management practices like cover cropping and nitrification and urease inhibitors. It also informs best management practices, as I said, like the 4 R's and cover cropping. And this study shows that we need more long-term studies, field scale studies determining how these management practices impact greenhouse gas emissions, both nitrous oxide and carbon. As you could see from Jacob's talk, our two researches kind of complement each other. Finally, I would like to acknowledge all the people in the EGMET lab, Dr. Claudia Wegner-Riddle and Sean Jordan, who really helped make this work happen, as well as the Create Climate Smart Soils program, University of Guelph, and NSERC. So yeah, thank you so much.
Wonderful, thank you, Nicole. And we have a few minutes for questions. We have about 3 minutes for questions. So I see that Yinsu has a question here. If the crop yields, are the crop yields different between treatments with and without NUIs?
Yeah, so I'll just quickly go back to that. Oops, too far. Quickly go back to that so you can see, because I know I went pretty fast. So we did see, as you can see here, we did see slightly higher yields in corn with the nitrification and urease inhibitors, 11.8 and 11.9. And we did see more, and we saw less variability within those yields as well. So, yeah, I guess that's your answer.
Great, thank you. And then another question is, how expensive are the inhibitors?
So that's one more thing that I want to, hopefully, if I have time, add into my discussion, into my thesis, is kind of a cost benefit analysis. Like, how is it, like, are the inhibitors affecting your yield? Are they really giving you that bang for your buck? Short answer, I'm not 100% certain on the cost right now. So I can't answer that, but I'm looking into it, because I do believe that just because these yields don't show we're not seeing significantly statistically significant differences between these yields, but statistically significance isn't necessarily significant to the farmer. They're going to feel those difference in yields. So yeah, I'm very much aware of that.
Great, thank you. And then a question from Aspen. Long-term experiments are expensive. What is your suggestion of conducting long-term experiments?
So yeah, long-term experiments are very expensive, and I understand that. But this site that we have has been a micrometeorological site for 20 years now. So it is a long-term experiment, and now that we are including nitrification and urease inhibitors for the next 2 growing seasons, we'll have a full rotation. You'll have the full cropping rotation in, and you'll be able to see the carbon and nitrous oxide exchange. So look out for those studies. We'll have those out soon. But my suggestion for conducting LTEs would be funding. So yeah. Thank you. Yeah, thank you so much for the opportunity.
Great, thank you for your talk. Excellent. So we'll move on to our talk by Wayne Hawking from the University of Western Ontario, and shifting gears a little bit in our subject area, but excited to hear the talk.
Okay, is that changing screens? I haven't got the wrong one or something. Can you see that? Am I changing slides?
Oh, no. Try, I still see just the slide deck.
Got the wrong one again. Okay. It's got to be there. Sorry, try again. Well, is this the one that works? Does that work?
Yes, perfect, thank you.
That works, does it? Good. All right, had the wrong one. Okay, so now for something completely different. The previous talks and most talks here are about chemistry and agriculture and land and such like interacting through anthropogenic effects. I'm talking about the effect of land and oceans and land-ocean interfaces in the sense of impact on the weather independent of any anthropogenic effects. And you may say, well, we're all familiar with that. You know about lee waves, you know about mountain waves, you know about sea breezes and such like. So what's new? And I think what's new is quite interesting. So I'm showing here a pattern of clouds which represents what we call a gravity wave. It's a big structure which was seen at Mark's Day last year. And basically what these are a breakdown of the wave by Kelvin-Helmholtz instabilities. It's a very large structure. These things are quite rare. And so they attract attention when you see them. But the question is, how important are they long term? I've got some other examples here. This is you can see the structure here. This is on a cloud in Antarctica. You can see some banded structure up there, which could be gravity waves, and another picture here, which also can be gravity waves, and it's highlighted by the condensation associated with the structure of the wave. So you can see this banding structure. And so the question is, okay, how important are these? The general feeling is that large-scale turbulence, when I'm talking turbulence of the scales on kilometers and more, are the dominant process by which diffusion and such like happens in the atmosphere. Waves are, in some quarters, considered to be a relatively minor, but a significant but relatively minor contribution. There's another school of thought that says these waves, when there's enough of them, make a huge impact. And so these waves I've shown you here are visible, but there could be lots and lots of waves which are much smaller, but which and essentially invisible to the eye, but which can have quite dramatic effects on the weather. And so the question is, what's the balance? What's the balance between the waves and the turbulence, and how do they differ? And can we parameterize and determine in some way what effect they might have? Because this will have big importance in understanding the nature of weather systems and such like in the long term. We used what's called a wind profiler radar to measure the winds in the atmosphere from the ground level up to about 13 kilometers height over a period of about 15 years. And we have performed spectral analysis to do this. Now, what we do is the radar changes its beam direction from minute to minute, or roughly every 30 to 40 seconds. And each period we take one recorded average and we measure the radial velocity of the wind. From that, we can convert it to large-scale wind motions. And then with the time series, we produce spectra. And we're going to look at the nature of the spectra. What does it say about, what's theory say about two-dimensional turbulence and waves? Two-dimensional turbulence at these scales, and we're talking scales out from tens up to thousands of kilometers here, have a very detailed, very clear structure. It has Now, the structures themselves are physical, but because it's blown over top of us by the mean wind, we can translate it to a frequency spectrum, which is what's done normally with the Taylor frozen turbulence hypothesis. It's a fairly standard technique, and it works at quite large scales, not just small scales as well. You've got to be careful, but it does work. The basic structure is one of a slope in the spectrum of around about omega to the -5/3, which changes to omega to the -3, omega to the -3 at very, very large scales. And this is fairly well known. And the effect of Doppler shifting, motion of the wind, is just to slide the whole thing to the left or right. The shape stays the same. And this is the region I'm going to be interested in. This is what we can study with. So this is going to take us from periods of about 5 minutes out to periods of about 20 hours. So we're going to be looking at this structure. On the other hand, the structure for turbulence for waves is quite different. There are multiple different ways in which waves form, and I've shown classes of them here. The most important ones to us are this green region, which are internal gravity waves. There are also things called external surface waves, mixed modes, planetary waves. Planetary waves are scales where the wavelength is comparable to the radius of the Earth. You might get 10 oscillations around the Earth. So very large systems, essentially the size of a high or low pressure system. And you'll notice the one thing about them, this has an omega to the -5/3 law, a frequency to the -5/3, which is exactly the same as the law we had for the two-dimensional turbulence. So you may say, well, I can't distinguish between them. Now, I will point out to you, when we want to look at these waves, you can look at them in the sky, but when there's so many of them, a whole bunch of waves working together with random orientations looks a lot like turbulence. You think of them flying over the ocean, you've got a lot of waves sloshing around all over the place in the middle of a storm. You can't tell whether they're waves or turbulence because there's so many of them interacting together. So you can't just look and see, you have to actually do experiments. So the spectra are the same, so in principle, but that's what we would call something called the intrinsic spectrum. That's the spectrum the waves would have if they were not moved around, changed by the Doppler shifting of the wind. It turns out this mean wind motion and Doppler shifting makes a big change in the spectrum. Why does it matter? It matters because waves can force the atmosphere in ways that two-dimensional geostrophic turbulence cannot. Think about just waves washing up onto the beach. Coordinated wave motions can end up washing the sand from one end of the beach to the other. Turbulence just sloshes it around back and forth. So waves working together, even if small, can produce very dramatic effects on larger scale systems. And this is why it's important. So this is the network of radars we used. All these little X's, they go everywhere from Harrow over to Montreal and up to Oman. And there's also another one we have in Eureka in the Arctic. And each of these do the same thing. This is a picture of a radar. There's a large field of antennas covering typically 50 to 60 meters across. We use this to form a narrow beam and point the radar signal into the atmosphere. It shows a side-looking view. You can see we form what we call a narrow beam here. This might have a width of 2 or 3 degrees. We point it into the atmosphere. The motions of the turbulence and the scatterers moving overhead allow us to then measure the wind speed. If you look from above, we can point this to different directions in the sky. So here I pointed to the north, here I pointed to the east, and so forth. And we do that on a regular basis for weather forecasting. So we have a very fixed cycles that we use for this beam pointing. This gives us a problem for what I want to do, though, because what I want to do is actually take one of these beam positions and form time series over 20 years of data. So I've got to be selective of which beam I choose. This means that when I choose the beam, and I'll say I choose the north beam, my interval between recordings on any one beam is typically 5 to 15 minutes. So it's not 1 minute. It'd be nice if it was 1 minute, but that would mean we'd have to point the radar in one direction forever, and we wouldn't get all the useful weather information. So to compensate for that, we use something called a data compensated discrete Fourier transform, which allows you to take Fourier transforms and produce spectra with data with non-equally spaced data points. And that's what we've done. And just back to the theory then, remember what we're doing here. The turbulence predicted a K to the -5/3 power law, which becomes a frequency to the -5/3 power law when we do the studies with the radar. And gravity waves predicted omega to the -5/3 as well. But again, that was what we call the intrinsic spectrum. That's the waves which is embedded there, where we just look at each wave individually and forget about the fact that they all interact with each other. So there are ways to resolve these. One's momentum flux I won't discuss. The other point is the atmosphere is in continuous motion. It's being blown by the mean wind. This produces Doppler shifting, which changes the frequencies. Also, different scale waves act as Doppler shifting agents to other waves. So a large scale wave will act as a Doppler shifting agent to a smaller scale wave. So when we take that into account, the intrinsic spectrum that I showed you earlier, the one which basically says, let's forget about the waves talking to each other, has the -5/3 slope. But, well, I've showed you the turbulence plot. Even if you've got mean motions, the slope stays pretty much the same. It just slides back and forth depending on the mean wind. But the gravity wave spectrum changes dramatically. And in this region here, between about 5 minutes period and about 20 hours of period, depending on the mean wind and depending on how the waves interact with each other, the slope changes enormously. You can see when the green curve here, when there's no mean wind, is shown here. And yet by the time we get up to the point where the mean wind is substantially larger than the phase speeds of the waves, the slope becomes very, very flat. So in this region, we have the potential to identify wave action in ways that has never been done before. What we do is we take typically a month of data, we Fourier transform it with the DCFT. You can see 3 areas. This region gets kind of flat. This is where what we call the Taylor frozen turbulence hypothesis breaks down. We can't use that. This is noise associated with the fact that we have to select data with much longer period spacing between the points than we'd like. But this region in here is obviously got a slope and it's statistically significant. And I've shown 2 examples here just to show how it varies. Although there's a lot of noise, the slopes can be found. The correlation coefficients are typically 0.4 to 0.6 with these. So they're very definitely significant. So we select these regions out, fit the slopes, and we do this every month over a long period of time. So here's some data. We've got 4 years of data here for 3 of the sites. We've got all the data from 2010 to 2013. Once every month, we take a full monthly data set and do these Fourier transforms, and we plot it as a function of height and time. You can see the difference. Harrow and Walsingham are very red. That means the slopes are very shallow, which means they reflect high gravity wave activity. The one up higher is Wilberforce. It's got a lot more blues and greens in it, and only reaches the yellows. This is a site. So what happens is this is a site which is sort of in the middle of Ontario, some hills, but not a huge amount. We don't expect too much wave activity there. But Harrow and Walsingham are both very close to Lake Erie. So when the sea breezes blow in, they produce wind shears, which produce gravity waves. And so this is a region of highly active gravity wave structure. And so we can see from this technique, we can identify regions which have strong gravity wave activity from regions which do not. The next graph basically shows all of the sites. Walsingham and Harrow down the bottom, they're the ones closest to the lakes. McGill, quite a lot of red. That's because McGill has several mountainous areas around it, and also some very deep valleys with rivers flowing into Montreal, which produces a very complex structure. That's why Montreal has a very, very complicated weather system. And so that interacts with the synoptic flow above and produces large wind shears. So when you see these events, you can explain them, but in advance, you can't say for sure what they are. So each of these sites has different characteristics. And the point I want to make here is that each site, we can now say, are gravity waves important or are they not important? Now that's important for large scale numerical modeling, because the large scale, even the best GCMs, the best global mechanical models, have scales of maybe 100 kilometers as their resolution. They can't get down and see these little gravity waves. They can't model them. And if you try to do that for every site in the world, the problem just becomes enormous. You can't get down to that resolution. So what this does is tells you, these are the sites I should look at. So I should run a site, a numerical model at some local sites associated with Walsingham or Harrow, wherever this is important. And I can use that to parameterize the wave activity and then feed that into the larger GCM. And I don't have to waste time spending time looking for gravity wave activity in areas where it's less common. So this is early days, but this paper, this work has been very constructive, and it helps us distinguish waves and turbulence in the atmosphere, and that's been its purpose. So the conclusions are simple. Gravity waves can and do dominate at various sites and at various times. The idea that they're totally unimportant or rather a minor contribution is not true. Two-dimensional turbulence is more pronounced at lower altitudes. Gravity waves take over at high altitudes, and that's because gravity waves grow in height as they go up. By the time you reach the mesosphere, which is where I do a lot of my work as well, they are the dominant forcing mechanism. Gravity waves drive the entire circulation of the mesosphere. We just want to know how important are they down here at this level, and we would say they have some significance. A lot of site-to-site variability. There are gravity wave hotspots that we can focus on to improve numerical models. And I guess that's the main point. If people are interested, it's a paper here. You can take a screenshot of it if you like, which was presented in the Quarterly Journal of the Royal Meteorological Society, where we expand on all this stuff extensively. But I just wanted to make the point that waves in the atmosphere, even if they're tiny, when they work together in a cohesive manner, can have very significant effects on the prediction of weather forecasting. And so that was the point of this paper. So we're finished. Thank you.
Well, wonderful. Thank you very much for that very interesting talk. All right, so we have time for one question here. We have a question from NN. Both numerical weather prediction models and climate models have gravity wave parameterization. Has their resolution increased? A larger part of has their resolution increases, a larger part of gravity waves will be resolved.
Right, but as you want to have the resolution, you've got to have many times more time. There's a trade-off there. I don't think we've reached the stage yet where we can incorporate all gravity waves in there. A lot of GCMs do not even go above the tropopause, which is a problem in itself. So I think just recognizing these waves is important, and I think we're not yet at the stage that the smaller scale waves, waves with a scale of 5 or 10 kilometers or 20 kilometers do not get integrated into the models very well at all. And I think that they can be the most active triggers of storms and convection. So I don't think the models are yet able to take them and generate them internally. I think having this knowledge will offer a shortcut until we can ever reach that stage.
Wonderful, thank you so much. We'll move on to our next. So our next two are going to be poster presentations. So there'll be 5 minutes, and then if you have any questions, I encourage you to go discuss any of your questions that you have in their poster discussion session. So that's at 11:45 tomorrow. So yeah, if you have any questions related to their posters, that will be a great chance to connect with them. So our first poster being presented is from Kyla Wicks. So please go ahead and share your screen.
Hi, thank you. Okay. Are you able to see the poster? Okay, can everyone see the screen?
Yes. And if you could speak just a little bit louder, that would be great.
Louder? Okay, sorry. Hi everyone, my name is Kayla. I am an incoming master's student at the University of Guelph, and today I'll be presenting my undergraduate research entitled Controls of Deferring Non-Growing Season Cover Crops on Winter Soil Temperatures. One of the largest anthropogenic sources of nitrous oxide are cropland soils, and fluxes are associated with the amount of freeze-thaw cycles and total number of frozen soil days. Notably, winter cover crops influence soil temperature by insulating the topsoil below, and winter cover crops are also associated with reduced post-harvest soil nitrates. The main objectives of this study was to highlight soil temperature temporal variability as a result of specific cover crop alternatives through field observations, as well as understand how well soil freeze-thaw cycles are represented in the simulated heat and water model, or SHA for short. And moving on to methodology, our experiment ran from November of 2019 to March of 2020, so single non-growing season at the Elora Research Station in Aris, Ontario. Temperature eye buttons were inserted at a 5-centimeter depth into the topsoil of 25 locations across the experimental area. And the area included 8 different agricultural covers that were well established prior to the installation of the eye buttons. And these plots were about 1 meter by 1 meter in size, square, and they're illustrated in figure 2. Each of these covers, their average compositions were recorded to evaluate the percentage of bare soil, residue, and cover crop. And this is in figure 3, just down here. So our residue dominant treatment was no-till corn. The bare soil dominant treatments were tilled soy, corn, and fallow. And the vegetation treatments were a 3-mix, a 10-mix, oats, and red clover. The Shaw model was used to simulate soil temperature under each of the 8 covers using local hourly weather data, and input files were adjusted as needed based on cover composition. And these input files are in this schematic just over here. So something that was bare soil dominant, like the no-till corn, would have underlying soil node input files as well as a snowpack. In contrast, something that had vegetation, such as the red clover, had vegetation, snowpack, residue, and the underlying soil input files. We quantified the number of freeze-thaw cycles as any time the soil fell below a 0-degree threshold and cave above, that was considered 1 freeze-thaw cycle. And to get the total sum of frozen soil days, any day where the average temperature was below 0 was considered a frozen soil day. With regards to our field observations, the total number of freeze-thaw cycles and frozen soil days were significantly reduced under cover crops, specifically oat and 3-mix covers and residue-heavy treatments. However, when we ran the simulation using Shaw, freeze-thaw cycles were underestimated among bare soil dominant covers and overestimated for most of our cover crops, with the exception of red clover. The total number of frozen soil days were significantly overestimated for both vegetative and residue dominant covers, while in contrast, something like tilled soy and corn treatments were slightly underestimated using Shaw. The main conclusions of this study was that both winter cover crops and residue influence soil temperature by reducing the frequency and extent of soil freezing, insulating the soil below. However, much of these processes are inadequately represented by the simulated heat and water model. At this time, I'd like to thank Jason, Claudia, and Aaron for their ongoing support with this project, as well as Kajenra, Spencer, and Swapan for their assistant data collection, and my lab mates for offering their suggestions on the design of this poster. And lastly, the funding for this project was provided by the Natural Science and Engineering Research Council Canada and Canada First Research Excellence Fund. Thank you all for listening to my presentation.
Wonderful, thank you, Carla. And if you have any further questions or would like to hear more, I invite you to go by her poster during tomorrow's poster session from 11:45 to 12:45 Central Time. Wonderful. So up next for another poster presentation is Sibley Duckert. And please go ahead and share your poster.
All right. All right, just trying to find the presentation. Sorry. Oh, here we go. All right, so my name is Sibley Duckert, and I'm this study was a part of my undergraduate research at the Claudia Wagner Riddle Lab. And what I did was I performed a sub-analysis of the data set on net ecosystem exchange measurements of through eddy covariance methods to find the annual carbon budget of a cornfield in Elora, Ontario from 2012 to 2018. So the objectives of the studies were to quantify the long-term variations of the annual carbon budget across the 7 years, and also to examine the impacts of growing season length on these fluxes by comparing them to other cornfields across Northern America. So moving on to the next slide here, what we found that was when we considered the carbon removed from the harvest, all years except for 2012 were actually a net carbon source, which is obviously something we don't want. So if we look up in the top left corner here, we have a graph depicting the net ecosystem exchange, the daily net ecosystem exchange of the whole 7 years. And anything above 0 is a carbon source, and anything below is a carbon sink. And then the table below depicts the actual annual values for each thing. So we can see that the net ecosystem exchange is in the negatives for the first bit, but once we consider the carbon removed, so the net ecosystem carbon budget, we see that everything kind of turns positive except for 2012, indicating that it's actually a carbon source. The data was then compared to studies of cornfields in different latitudes, some further up north in Manitoba and some further in south in like Iowa, Nebraska, and Illinois. And what we found from this was that the net ecosystem exchange actually was more of a carbon sink when the growing season length was longer and when the air temperature was higher. Then we also compared the studies with their NECBs, so when the carbon was removed from harvest. And what we found here was that there was actually a relationship between the ratio of net ecosystem exchange to yield, and which was associated with a negative NECB with ratios above 50%. So that being said, the goal is to increase the net ecosystem exchange to result in carbon sinks. And this could be done through cover cropping or residue retention. However, further studies need to be made for that. And again, thank you to the entire lab group for helping me with this study.
Wonderful, great. Thank you for that presentation. And again, if you would like to speak, if you'd like to hear more about the poster, again, I encourage you to go to the poster session tomorrow. Great. So we're gonna have 3 more talks as part of this session. So our next speaker is gonna be Philip Carter from the Center of Hydrology at the University of Saskatchewan.
Thanks. Yeah, so I'm a research associate at U of S, and I'm going to be talking about coupling crop growth and cold regions hydrology through observations and modeling on the prairies. So shifting a little bit to the west. And there we go. So the main thing to think about when we talk about agriculture on the prairies is that it's really big. It covers 50% of the prairie ecozone, which covers a large portion of Alberta, Saskatchewan, Manitoba. And it exists in a place where we have fairly extreme climate. So we have cold winters with snow and frozen soil infiltration processes. And then in the summertime, it's hot, dry, and tends to be water limited. It's quite an interesting place in that we have a lot of variability. And the one thing that I like to point out is that if you look on the bottom right figure, the blue line being precipitation observed in Saskatoon historically and into the projection to the future with the uncertainty bounds there. And then red, looking at Saskatchewan spring wheat yields, we can see that there's a correlation between the amount of precipitation and wheat yields that happen interannually. But at the same time, we have this huge increase in yields. It's a tripling in yield since the early 1900s to current. And so this really emphasizes how adaptive Canadian prairie agriculture has been to successfully manage and grow in such an extreme climate. The challenge, though, is that if we're going to go into the future, we want to have some modeling capacity to understand what are the likely outcomes. And at this point, crop, water, ag interactions tend to be a little bit out of date. We haven't really appreciated a lot of the impacts that zero tillage has had, for example. And so conceptually, when we want to think about crop, water, ag, and you want to boil this down something to a very simplistic perspective, we're talking about dryland agriculture with zero till. And superimposed on this, all the cold region hydrologic processes. So in contrast to more temperate regions, there's this very large feedback between winter and summer, which I'm going to hopefully try and elaborate on here. So if we want to go throughout the year, wintertime occurs, we have snow accumulation and a snowpack, and a big factor in that is how well our crop stubbles are able to retain snow, and therefore will determine how much snow can infiltrate in springtime, which leads to what is the soil moisture condition at the start of the growing season. And then when we get into the growing season, typically, and this is not always the case, this year being an extreme example of all endpoints on the climate spectrum, but typically all available precipitation and soil moisture is used to determine how much evapotranspiration occurs and therefore crop water growth. So this is this framework that we work in. And so what we want to do in this work is, number one, we want to get some field-based observations to quantify these interactions that reflect current practices. And then number two, develop an agrohydrology model that can actually simulate some of these impacts, management options to understand prairie hydrology and climate moving forward. So starting with the observations, this is worked through the Global Water Futures Program, Ag Water Futures Project specifically. And so since 2015, we've been collecting data at a number of sites near Saskatoon. And the main focus of them that I'm presenting here is quantifying sort of the growing season crop water balance. So if we look at the growing season as a whole, generally, evapotranspiration is equal to precipitation plus a change in soil moisture, because prairie soils tend to have very high infiltrabilities and have limited runoff. So we want to try and close this water balance as best we can with independent observations. So using evapotranspiration from anticovariant systems, precipitation with networks of GNORs and tipping buckets, and looking at soil moisture with a whole suite of hydroprobes for near surface, getting into neutron probes to get into the deeper surfaces. And we've got, I think, in total about 19 site years now for sites near Saskatoon. We've also collected a bunch of other data to go along with it: energy balance, biomass, leaf area index, soil properties, and not the focus of this at all, but also extensive spatial UAV data with LiDAR, infrared, RGB, multispectral, and getting into gamma spectroscopy to try and describe some of the spatial aspects. So when we go out, collect all this data, gap fill, energy balance, closure on our evapotranspiration, what we end up having is these growing season water balances. So red being crop water use as captured by our ecovariance, blue being the rainfall that we've observed, and green being the change in soil moisture over the course of the growing season, from seeding to harvest. First thing you want to take note of is that we're in water limited situations, and so growing season evapotranspiration can be as little as 150 millimeters up to 400 millimeters, and this is in contrast to potential evaporation values often that we estimate to be around 700 millimeters because of the hot, dry conditions. If we take on average all of these components, we can see that for these sites and these times, 62% of evapotranspiration can be accounted for by the rainfall that's observed. 12% can be accounted for by the soil moisture differences that we see, and that leaves us 26% residual, which is unsatisfying to have, but it is consistently a gap where we're not a negative in the equation. And so what this does tell us is a couple things. One is we've got a scale mismatch between our evapotranspiration and our point scale of soil moisture relative to our covariance footprints. And the other aspect is that prairie hydrology is quite spatially variable and depression focused. And so we're really not capturing a lot of the dynamics in our lower parts of the landscape that are generating most of the crop growth and evapotranspiration. So there's some gaps in there. What we can say though, with more confidence, is that 38% of the crop water use came from non-growing season hydrology for these sites. And so winter hydrology is really important in determining crop water use come the summertime. And if we look at the wintertime water balance for the periods where we have good enough data to do that, you can start considering things like efficiency. So how efficient are we able to retain snow in our landscape? On average, it's about 0.5 of 50% of our snow is retained on the landscape and available to melt. So this is peak snow water equivalent divided by snowfall observed over the winter. And then if we look at soil moisture retention efficiency, so this is the amount of soil moisture change relative to snow that we've observed, peak snow plus rainfall, it's actually quite low as well. So what I'm trying to communicate here is that we have opportunities potentially to increase the amount of water available for crop growth in the summertime by managing what happens in the wintertime. But to do that, we need to have a modeling framework to try and relate these things, because we can't measure everything all the time. So this is where we've been developing the process-based ag model. I'm calling it the Cold Regions Agrohydrology Model, because CRAM, because everything needs an acronym. And this is basically a coupling of the Cold Regions Hydrological Model, which simulates cold region hydrology, snow processes specifically, frozen soil infiltration, and coupling this with an aqua-crop OS, which is a water-limited crop growth system. So we can set it up in sort of a loose way and pass things back and forth and start simulating things like stubble height impacts on yield in summertime. And so, but the first thing is aqua-crop OS has not been used all that much, so we need to validate it. So from a subset of these, we can start looking at how well we're representing biomass, evapotranspiration. Keep in mind, 2021 was kind of a terrible year, so take that with a grain of salt, but we're doing pretty good. And then we can couple this together, and then conceptually, we can start trying to simulate a stubble height crop growth response. So in principle, if we increase stubble height, we're going to increase the amount of snow available because we're reducing sublimation and snow transport, which will increase the amount of snow melt, which increases soil moisture recharge and runoff. And then ultimately more soil moisture will give you more crop growth. And then, so putting this in, trying to put some numbers to it, first off, this is a subset of data looking at specifically barley from 2018 to 2021 and looking at 3 different stubble heights, 5 centimeter, 25, and 50 centimeters. Very quickly, we see things are non-linear. It's not just you increase stubble, the things that increase in the exact linear response kind of way. First off, the primary response is runoff, more snow, more runoff. But we also see that biomass does increase. So if we increase stubbles from 25 to 25 centimeters, we're increasing our yields between 3 to 6%. 5 to 50 centimeter stubble heights will give us a 10 to 32% increase in biomass, which is analogous to yield. So in principle, we can make this work. And this is just in terms of biomass as well. So in summary, we really need to start relating crop water interactions from a water year perspective to couple what's happening in the winter with what's happening in the summer. And so we've got a tool now to do this with CRAM, and you can start simulating ag practices. And with this, sort of the next steps we're going to go with it is, first off, we need to start doing better at soil evaporation post snowmelt and accounting for residue differences. We need to start looking at biomass stubble height crop residue feedbacks. So we had a drought last year and there was not a lot of biomass produced. Therefore, we can't have tall stubble heights to get us through in terms of the snow retention. We also need to start thinking about the spatial variability of depression-focused hydrology. And what we're going to be doing as we resolve some of these questions is going to be running it over the Canadian Prairie Ag extent to look at some of the impacts of climate change on crop water use and productivity. And then as well to start looking at some stubble and surface management practices that we can use to try and optimize crop water use efficiency to try and address some of the resilience questions as we move forward with climate change rearing its head. So I'll stop there for now.
Wonderful, great. Thank you so much for that talk. And we have some time for some questions. All right, so from Claudia, what is the typical height of stubble and what is its function, e.g. combined used crop, and how much opportunity for adjustment?
It depends on what could grow. So last year we had very short crops because of drought. And I would hazard to say that you probably couldn't have any stubble higher than 20 centimeters in most parts of the prairies. But regular conditions, currently 15 to 20 centimeters is often what you see. But there's also opportunities in terms of what we call stripper headers. And so this is where you have a straight combining situation, and what's only being removed, it's a stripping motion that kind of just pulls the heads off your cereal grains, for example. And so you can have stubble heights, however tall the crop grew, up to 75 centimeters in a very productive area, for example. So there is a huge range that you can play around with, but it really depends on equipment and what your farmers are able to manage. Yeah.
Great, thank you. And then we have a question from Catherine. What is the minimum spatial resolution the model would need to fully capture the spatial variability of depressions in this region?
Great question. At this point, this model is a 1D scenario. And so to try and capture depression focused recharge and that kind of stuff, you're gonna have to get into some more clever modeling to get into some of those 2D relationships. In theory, it depends not so much on what the model is right now, but how you set it up in terms of your forcing data and how we represent your soils and stuff. And are we using, spatially explicit distributed modeling processes, or are we starting to talk about HRU based stuff, which is what happens in CRIM model, for example. To capture a spatially variable hydrology, you have to go probably, Landsat scale, for example. Some depressions can be quite small.
Wonderful, thanks. And Aston has a question. Maybe I'll let you two connect directly for this question, just so that we can help stay on time.
Yep.
But yes, thank you for your talk. And we'll go back to the University of Guelph, where we have a talk from Ryan LaFleur.
Hey, I know this doesn't land as it does normally in person, but I really like your shirt there, Philip. I think it's, we'll keep going with that color. But yeah, so let me see. Oops, share screen. Yeah, that one. Okay, we should all be seeing presentation view. And then I can look at the camera and my screen. Looks good. All right, so today, so just to reiterate, my name is Ryan LaFleur. Today, I'd like to keep the ball rolling with winter water and talk about winter water budgets under altered freeze-thaw cycles. So this was a lysimeter study that we conducted at the research station, at the Elora Research Station, just north of Guelph, Ontario, Canada, which is in a humid continental climate. So just to reiterate some of the points that some of our other speakers said and some background on my research, it has been noticed that over the past 50 years or so, there's been about a 5 to 10% decrease per decade of the what's called the snow cover fraction, which just refers to the thickness and the duration of the snowpack throughout the non-growing season. Other research has also suggested that this idea of these warmer waters through the action of reducing this, warmer winters through the action of decreasing the snowpack can in fact induce colder soils and in turn potentially alter water budgets in the soil. So this is something that we found pretty interesting, and we figured that this would be pretty important to study and maybe worth simulating different winter conditions in order to observe what happens to the water budget. So what is a freeze-thaw cycle? So here I have a couple examples, but a freeze-thaw cycle basically is simply a period of freezing or sub-zero temperatures followed by a period of above zero temperatures. And what we wanted to do for our experiment is we wanted to create two versions of a warmer winter with less snow, where one has, so here's my treatment, where one has little to no freeze-thaw cycles, giving it warmer soils. And then the second here has enhanced or more more freezing or freeze-thaw cycles, so giving us a warmer winter with colder soils. So the objective of our assessment was to assess the impact of these two different simulated winter conditions on the water budget, primarily its outputs and water storage. And we wanted to do this on two different soil types, one being a silty loam and one being a loamy sand. So before we get into any results, I wanted to talk a bit more about what exactly the water budget is. So the water budget is a simple summation of all the inputs and outputs of water through your volume of soil of interest. So here you can see that our inputs are things like precipitation, whether that precipitation be snow or rain. And then we have our output. So what we did here was we combined evapotranspiration and runoff into something we call surface water loss. And then we have our subsurface water loss, which is our drainage. And then if you subtract your outputs from your inputs, you should get whatever water is left behind in your soil, right? And this is the part that's very important for the following agricultural season. So here it is. This is the Elora Research Station, the lysimeter site, where basically we have these 3 hexes, each with 18 meter squared lysimeters that are on these weighing scales. And they're also hooked up with a bunch of temperature probes, matrix potential probes, water content probes, you name it, it's got it. One of the things that I found really cool about these is that these are actually intact soil cores. So these were pounded in and then dug out from around in order to keep all the soil in there perfectly intact the way it was in the field where it was retrieved. So here, just to give you a bit more details on the site and our experiment. So here we wanted to see about how it would work with a imperfectly drained silty loam, so not very good drainage, and then a well-drained sandy loam, right? So 2 different spectrums, 2 different ends of the spectrum for drainage. So as you probably noticed, the site is fairly large. So what we did in order to average in some of that spatial variability is that we have 1 control and 1 treatment lysimeter for both soil types in each hex, just to cover over that distance. No need to worry about the non-diverse because all of the ones that we used are under the diverse management treatment, crop management. All right, and so what we ended up doing was we applied these ceramic heaters that you can see here and here. And so for example, for the continuous warming treatment, we just had them on all winter long. And then for the intermittent warming treatment, we tried to time the melting of the snow with the natural cold fronts that come through during the season to try and let it warm up and then have no snow, no insulation as the cold came through. So in my results, I'm going to be presenting to you 3 different seasons. The first one being a no treatment season, just so that we can have a good baseline to see how the 2 groups or how the 2 triplicates of lysimeters compare to one another before any treatment was applied. Our second one is going to be the continuous warming, and then the third is going to be the intermittent warming. So let's get into some results. First thing we want to check for this type of experiment is that whether or not we actually achieve to create a warmer and colder soils. To start off here, we have our no treatment season where we have good agreement, good behavior between our 2 groups. And then for our continuously warm treatment, we see that we did in fact create a significantly warmer soil throughout the entire season, little to no freeze-thaw cycles occurring in both soil types. And then for intermittent warming, we managed to cool down by heating intermittently. We managed to create very significantly colder soils than their accompanying control group. So that already for me was already a pretty awesome success. So now to continue, now it's like our observation, now we want to look at how exactly did this affect the water budget inside the lysimeters. So to start off, we want to look at storage. How much water actually was able to be stored if there was less. So here we're seeing that the continuous warming treatment in both soil types did cause a significant reduction in the amount of water that was able to be stored by the end of the season inside the lysimeter. However, our intermittently warmed didn't quite fit the bill. So it didn't quite do much. So there was no real significant differences to report. So now where did that water go? So we have a couple options. We have our surface water loss and our drainage. So to start off, let's try and see if we can find it in our drainage. And here, one thing I'd like to note is that here we have good agreement before any treatment. However, in our sand, for whatever reason, there actually was a bias. Our triplicate for our warmed group on average before any treatment was applied did have a significantly more drainage than its control counterpart. However, even with this bias taken into account, our continuous warming did increase, did make a bigger gap between the control and the warmed groups. So there is a significant increase in drainage in both the loam and the sand lysimeters. And then, as we saw previously, there wasn't really change in water storage, so we don't really expect any large change in the drainage for the intermittent group. Lastly, just to check we're going to look at our surface water loss. So here we're seeing good agreement before the treatment occurred. Our continuous warming didn't do too much, no real changes in our continuous warming for the surface water loss. And then our intermittent warming had no real changes in the sand. However, there is a small significant change occurring in the loam. So that might be something of interest later on. Maybe if we were to make a more intense intermittent warming, maybe we could have extracted that to be something a bit more significant. So to summarize it all up, we confirmed that warmer winters can indeed cause colder soils. A warmer winter with warmer soils can lead to a reduction in water storage in multiple ranges of drainage of soils. And basically that water, instead of being stored in the soil, is going to drain out the bottom and join subterranean aquifers or the water table below. A intermediate warming has the potential to reduce water storage by increasing surface water loss in poorly drained soils. And lastly, to wrap this all up with a bow, basically what we're trying to say is that depending on how climate change affects our warmer winters and how it removes or decreases the snow cover, we can see some very contrasting effects on water budgets in agriculture environments, forest environments, everywhere. Thank you very much. I'd like to acknowledge my advisor, Claudia Wagner-Riddle, my committee, Aaron and Asim, as well as Sean. He's the man who makes everything run on the day-to-day, Shannon and Zach, and the rest of the group. So thank you very much. Any questions?
Great, thank you, Ryan. And we have time for a question or two. Give folks a second to type in their question. All right, so from Yinsu, are the differences in overwinter snow, do the differences in overwinter snow amount play a role in the water budget of the treatment?
Sorry, can you say that again?
So do the differences in overwinter snow amount play a role in the water budget of the treatments?
Well, the assumption would be that like as in changing between the control and the warm treatment. So it would be like, the assumption would be that the control and the warm treatment are receiving approximately the same amount of snow. Obviously, there is some spatial variability present. However, using having one lysimeter in each hex for the warm and the treated, we should be able to average over any spatial differentiation like that.
I mean, sorry, I just mean between the 3 years of treatment, is there a huge difference between snow amount over the winter? If that will play a role.
Well, it would definitely play a role if we were to compare, say, like 2016, 2017 to 2018, 2019. Yeah, it would play a role there, and that's why and that's why we made sure we had a control and a warm treatment per year, because we knew trying to do a comparison from one year to the next probably wouldn't amount to much, because yeah, like you said, there's like a lot more variables than in how much precipitation occurs and when it comes in. If that answers your question.
Thank you.
Great. All right. So thank you, Ryan. We'll move on to our last but not least speaker. So Shannon Brown, also from the University of Guelph, and please take it away.
Great. Share screen. So can you see my screen? Yep, okay, great. Yeah. Okay, great. So yeah, today I'll be talking about a comparison of nitrous oxide flux measurements from flux gradient and eddy covariance systems. So we're going back again to agricultural N2O emissions. We talked about this at the beginning, but so we'll end with it as well. So overall, industry and government, they're looking for practices to reduce N2O emissions for recommendations. So these agricultural N2O emissions, they occur post-fertilization and during winter freeze-thaw periods. And these emissions are driven largely by variations in soil, weather, and management, and the emissions themselves are highly variable. So when it comes to measuring N2O fluxes, a challenge is that we do have these spurious events that are heterogeneous in time and then heterogeneous in space as well. So there's usually hot spots of emission in any given field that's emitting N2O. And then the timing of these emissions is also very unpredictable. So to capture those fluxes, it'd be ideal to have continuous, spatially integrated, and long-term measurements of N2O. And so another challenge is that these fluxes are dependent on weather and soil conditions. So if we're looking at things like comparing treatments or comparing different management practices, ideally you'd want to have this occur under the same soil and weather conditions. So that would require the place that is just some measurements in one location that has the same weather and same soil. Another challenge is that it's often cost and power prohibitive to make these N2O flux measurements. Analyzers for N2O are typically very expensive, and if you're looking at powering a site, you typically need line power for these analyzers. And so then if you were thinking again of treatment comparisons, it'd be advantageous to have just one analyzer. So most studies typically just have one. So the spatially integrated long-term flux measurements typically done by using eddy covariance methodology, where you have a high frequency concentration measurement with a high frequency wind speed. And then also the flux gradient method. So that's where a difference in concentration is measured with height. And then that's scaled by the degree of turbulence in the atmosphere. And that's an eddy diffusivity. So eddy covariance, it is a more direct measurement, and it's the gold standard of micromet methodologies. One limitation, though, is that you can typically just get one measurement per site. So you can't really get replications in using just one eddy covariance tower. And the flux gradient method, it's been around for a very long time, but it has fallen out of favor and fallen out of use in comparison to eddy covariance. And that's mostly because there's uncertainty from parameterizations that are used in the flux calculation. And it's also fairly sensitive to the site that you're applying the installing the instrumentation in. So it is more sensitive to effect requirements and tower height. But some of the advantages is that you can set up a flux gradient system in a multi-plot configuration using one analyzer. So you can get that 4 plots or 4 measurements for the price of one with this type of configuration. And another advantage of flux gradient is that you can still measure during rain events. So most sonic anemometers don't measure during rain periods, and this is important for N2O measurements because often those emission periods occur during rain events. And then also another advantage is that with the currently available instrumentation, there is potential to measure using a low amount of power. So the objectives of this study were to develop and test a low power flux gradient instrumentation, and then make comparisons of flux gradient nitrous oxide fluxes against those measured by an eddy covariance system. And this was done using both a low power flux gradient and a multi-plot flux gradient system. So this again is another study out of Elora. So this is where it's located in southwestern Ontario. So the study was, study area, it's a 30 hectare site that has four hectare plots within that aerodynamically homogeneous area. So the low power flux gradient system was operated from May to July of 2017, and it was installed in just one plot. So it was just a single measurement. And throughout the study period for this the system, the power use was measured. So the system consisted of 2 AP200 intakes from and so then what makes this system low power different from typical operation of TJ100A is that instead of a large pump that would be close to 600 watts, the air is being drawn from the intakes to the analyzer using 3 micropumps that use just 7 watts each. So because it's a flux gradient system, we don't need to have the same frequency or same response time as eddy covariance system. So we can use these low power pumps to draw air to the analyzer. So the eddy covariance system was co-located on that same tower, so right in between the 2 intakes of the low power system. And this operated from May 2017 to April 2018. And so this is so that we get some winter measurements using the eddy covariance system. And so now the multi-plot flux gradient system, it's been operating since 2000. and it's split into 4 plots. And in the year of this study, so 2017, there were 4 different fertilizer treatments, being one applied to each plot. So the system consists of an intake tower, one in each site, and then each of those intakes is connected to tubing that brings it to a valve manifold. And so the valve manifold switches intakes between sites, and then that switching determines which airstream is being drawn into the TGA-100A trace gas analyzer. And so that so this system is measuring both N2O and
Oh, Shannon. Sorry, it looks like you're muted and the screen share stopped.
there we go.
Okay.
Is it back now?
Yes. All right.
Okay, great.
There.
Yeah, sorry, I think my internet cut out there for half a second. So yeah, so maybe there was some things missed, I'm not sure, but we'll continue on. So results with the N2O emissions over that 2017 spring of 2017 period. There were 2 emission events. And so one was a smaller event after the first rainfall on May 22nd. And this was a very dry spring, so there was so the soil moisture was very low when that first rainfall event occurred. And then again, it was very dry. And then in June, there was a second larger rainfall event that triggered stronger emissions from the site. So all 3 systems were operating during this time, and all 3 systems captured the same dynamics of the N2O flux emissions. So the winter of 2018, there was only the multi-plot flux gradient and the eddy covariance system operating. And there was one spring winter, and that coincided with periods of precipitation. So in this case, only the flux gradient system was able to measure the thaw emissions. So that showed the advantage of having this alternate method of measuring fluxes as opposed to eddy covariance. So in comparing results, there was a very good correlation between the N2O flux measurements of the low power flux gradient and eddy covariance system. And this was all done with a minimal power use of the low power flux gradient system, with using only about 25 watts during that period. So overall, it was good results to see, no significant over underestimation, and again, that good correlation. Results for comparison of the N2O fluxes from the multi-plot system to eddy covariance were a little bit more mixed. There was a lower correlation and showed an overall underestimation of the fluxes measured. However, this was kind of split. There was the multi-plot flux gradient system measured higher emissions during that first flux event, and then lower emissions. So